Hello, this is video number 3 for Module 7. And in this video, I'll be talking a little bit more about the experiments of Francois Jacob and Jacques Monod about the lac operon. And the experiments I'll be describing are using, they're using mutant alleles that they recovered in various genes of the lac operon, and determining whether these allele combinations are, which are dominant alleles and which are recessive when you pair them together. Now, in order to do this type of work, it requires a little bit of a trick because E. coli is a haploid organism. It has only one chromosome and only one lac operon. So how do you get two alleles together? So I prepared a short video to describe how that's done. And then once we have that, there's another video which is going to describe the experiment to determine dominance and recessiveness. Okay, second set of experiments that Jacob and Monod did were genetic experiments. And they wanted to ask the question, which of the alleles that they discovered, or the mutant alleles that they recovered, I minus, Z minus, Y minus, the super repressor I super S, and the operator constitutive mutants, which are dominant and which are recessive. And so that presents a problem to ask that question in E. coli. And the reason is E. coli has one chromosome, and that chromosome has one copy of the lac operon. In order to check for dominance or recessiveness, you need two copies. Which allele is dominant to the other allele, okay? So you can do a trick in E. coli and in prokaryotes in general, and you can insert a small circular piece of DNA containing whatever genes you want. You can make this by recombinant DNA technology. Jacob and Monod did this with a very primitive version of recombinant DNA technology. And so they inserted a second copy of the lac operon on this small circle, the plasmid, and the E. coli has another copy on the bacterial chromosome, okay? So that's what I'm representing here in this diagram. They created what's called a merodiploid. It was diploid only for a small portion of the chromosome. Chromosome is much larger, and what they inserted, a plasmid called the F' plasmid, had another copy of the lac operon. And it also had another copy of the lac repressor gene. All right, so now this merodiploid has two copies of the lac operon. And if you have different alleles that you want to test, you can determine which is dominant and which is recessive. So they made 3 different strains of E. coli having a plasmid, having the plasmid and the chromosomal version of the lac operon. And these were the genotypes. So in these genotypes, we're indicating what is the genotype of the repressor gene, the operator, lac Z and lac Y. And a plus superscript always indicates wild type, functional, and a minus indicates mutant, null allele, okay? Here are the 3 combinations, and in all of these combinations, the first copy is going to be the plasmid copy, the F' copy, and the second will be the chromosomal copy. And in all of these combinations also, the plasmid copy has a mutant allele of the lac Z gene and a wild type allele of the lac Y gene. And the chromosomal copy will have the opposite, wild type lac Z, mutant lac Y. The reason for that becomes apparent in a second. Here are the tests that they did. If they have an E. coli that has one wild type and one mutant repressor, which of those would be dominant? Wild type dominant or repressor dominant, or mutant dominant? Okay. Second test that they did is one copy has the super repressor allele. The other has the wild type allele. Question, which is dominant? Okay. Third thing they did was wild type operator versus operator constitutive mutation. Which of those is dominant? Okay, so the way they did this was taking these strains and finding out if they express LAC-Z and LAC-Y at all. They should be able to because they have at least one wild type copy of each. All right. And if they express LAC-Z and LAC-Y, is it inducible expression, like in wild type E. coli, or is it constitutive expression? Okay, so that's the setup for the experiment. And in the next video, I'm going to walk through how the experiment was done and the main results that they received. So I've redrawn the 3 experiments that Jacob and Monod carried out. And here are the 3 genotypes that were in the previous video. And they're testing, does the LAC repressor, is the wild type dominant to the mutant? Number 1. Is the super repressor dominant to the wild type? Number 2. And how about the situation for operator wild type versus constitutive alleles? Okay, so how they determined the phenotype, I guess, was by testing for the activity of the enzymes. Are they present or not? So LAC-Z, beta-galactosidase, if you if you test these bacteria with lactose, are they able to break it down or not? Okay, so positive test is lactose is broken down. Negative, it's not broken down, meaning no LAC-Z is produced. Is LAC-Y present or absent, right? Same thing. And they did this in the absence of lactose and in the presence of lactose. Remember, for the wild type E. coli, neither of these are present in the absence of lactose. Both of them are induced and present in the presence of lactose. Okay? So here are the results that they got. When they used this merodiploid and they grew it in the absence of lactose, both LAC-Z and LAC-Y, even though they're expressed from different places, one on the chromosome and one on the F' plasmid, both of them were absent, minus, without lactose. Okay? And then when you add lactose, both of them got expressed. So both of them were present. All right, so this is just like the wild type situation. And notice that it doesn't matter whether the copy that you have is wild type for the repressor, or mutant for the repressor. LAC-Y can be expressed even though it's linked to a wild type repressor. LAC-Z can be expressed even though it's linked to a mutant repressor. Both genes, even though they're in different places, are not expressed without lactose and induced with lactose. Okay, that's one result. Second result is this. When you combine 2 alleles of the repressor, now the super repressor, and remember the super repressor can't bind to the inducer and it can't be pulled off of the operator. And you make a merodiploid with a wild type repressor. And otherwise the Z and Y genotypes are the same throughout these tests. Okay, what is the result for that? Well, for growing this strain in the absence of lactose, there is no production of LAC-Z or LAC-Y. And now remember, the super repressor can't be pulled off the operator. Is it true for both copies of the operator here? The operator is wild type, both in the plasmid and in the chromosome. What's the situation? Well, what they find is that, yes, it's true for both. And if you add lactose, there is no expression from the plasmid, even though the operator is wild type. And that's because this super repressor can bind to the operator, can't be pulled off. can't transcribe Z and Y. But also true for this, lac Z cannot be expressed. It's not inducible, even though the repressor is wild type and the operator is wild type as well. So wild type alleles of both. What that means is that this super repressor is binding not only to this operator, but also to this operator. It can't be pulled off either. And as a result of that, you don't get induction for either lac Z or lac Y. Okay? Third result, what if we compare a wild type operator with a constitutive operator? What do we see? All right, so in this case, in the absence of lactose, what we see is that lac Z is expressed, lac Y is not expressed. Absence of lactose. Lactose doesn't really matter for lac Z. It's going to be expressed anyway. And notice that the lac Z is linked to the constitutive allele of the operator, and the lac Y is linked to the wild type allele of the operator. Now, if we add lactose, what are we going to see? So you're going to see actually expression of both. And the lac Z is expressed anyway, even in the absence of lactose, and that's not going to change if you add lactose. The lac Y allele is linked to the wild type operator, and so adding lactose is going to pull the repressor off of the wild type allele, operator allele, okay? And then lac Y can be expressed, okay? Here are the conclusions from these experiments. So one of them is asking about dominance and recessiveness of the repressor. And what we can see is that the wild type repressor is dominant to the null repressor, right? And the reason we can see that is that this repressor is going to mask expression of the mutant repressor. In the absence of the mutant, in the normal, if this was not a diploid, if this was a E. coli with just this genotype, mutant repressor would result in constitutive expression of the lac operon. But now if you add in a wild type repressor, then this becomes inducible expression again, okay? So that means that the wild type repressor is dominant to the null repressor. Second conclusion, the super repressor is dominant to the wild type repressor. And we can see that because there is no induction for either allele. So this wild type repressor is masking you would see induction normally from a wild type repressor. The super repressor is masking that. You don't see induction. So the super repressor is dominant to the wild type repressor. Third conclusion is that the repressor is transacting. What this means is the repressor doesn't matter whether it's linked to the operon or not. The repressor, this repressor is dominant wild type, and it's going to bind to the operator on both of these operons. The one it's physically linked to, the plasmid, and also the one it's not physically linked to, the chromosome, right? And we know that because both lac Z, which is on the chromosome, the wild type version, and lac Y, which is on the plasmid, are showing inducible expression, all right? So this wild type repressor must be binding to both operators, and then in the presence of lactose, it's going to be pulled off both operators. That's what transacting means. It's a substance that's made and diffuses through the cell, and it can bind to either operator, okay? So the repressor is transacting, and you can also see that for this combination, the super repressor is going to bind to both operators, and it can't be pulled off either operator. And so adding lactose is not going to induce expression in the case of a super repressor. The final conclusion is that the operator is a cis-acting regulatory element. And how we know that is from the third experiment here. If you mutate this operator so that there's constitutive expression, and remember, this OC mutant doesn't allow the repressor to bind to it, so it can never be turned off. So this operon will always express, and since the wild type allele of LAC-Z is present, LAC-Z is going to be expressed in the presence or absence of lactose. However, this constitutive mutant is not acting on LAC-Y on the plasmid. It's only acting on LAC-Z on the chromosome. And the LAC-Y is under the control of the wild type operator. It's showing normal induction, right? Not expressed in the absence of lactose, expressed in the presence of lactose. Okay, so the operator is cis-acting. Cis-acting means it regulates only what it's physically linked to. Okay, so there are transacting regulators. The LAC repressor is transacting, and there are cis-acting regulators. Transacting regulators are diffusible substances. They can bind to regulatory elements, multiple regulatory elements in the cell. Cis-acting elements are not diffusible. They only regulate what they're physically linked to. And this is a theme that we'll continue with when we talk about eukaryotic gene regulation as well, trans and cis-acting factors. Okay, so I just want to go through a few slides to reinforce what those videos were about. This slide is showing the first experiment in which the F' plasmid carried a wild type repressor and the chromosome carried a mutant repressor. And here's what's going on in, in the cell. So the repressor is acting in trans. It can bind both to this operator on the operon in the plasmid and to this operator in the operon on the chromosome. So it's a transacting factor, is a diffusible substance. In this case, in almost all cases, it's referring to a protein that's made by the LAC-I gene, and this protein can bind to either operator, okay? So that's a transacting factor. And that's also seen for this experiment in which the super repressor is encoded on the gene on the plasmid, and the wild type repressor is encoded on the gene on the chromosome. And the super repressor, you get both repressors made in this case. Wild type, it can diffuse. Super repressor, it can diffuse. So when they're first made, both of them are going to bind to copies, can bind to both copies. In the presence of lactose, however, the inducer molecule binds to the wild type repressor and pulls it off. The super repressor, remember, is mutated in the inducer binding site. It can't be pulled off. In fact, when the wild type repressor gets pulled off in the presence of inducer, any super repressor around is then going to bind and occupy that site. And it's going to end up shutting off operons on both the plasmid and on the chromosome. Okay, so the repressor is a diffusible protein which acts to regulate in trans. And the final experiment here is showing the situation for the operator, wild type operator on the plasmid, constitutive operator on the chromosome, and the repressors that can be made from either copy can bind to the operator. They can diffuse through the cell and bind to either operator. Well, they can only bind to this operator, actually, the wild type operator. The constitutive operator, the binding site is mutated in the DNA of this copy. Neither repressor can act on it. So that means genes linked to this operator are going to be constitutively expressed. The wild type LAC-Z is linked to this constitutive operator. The wild type LAC-Y is linked to this wild type operator. So this one is constitutively expressed, this LAC-Z. LAC-Y is induced in its expression, off in the absence of lactose and on in the presence. All right, this slide is just a table, and this is pretty much what I drew on the board summarizing those effects. So I'm going to end this video here. See ya.